Bueno, gracias para la introducción y me gustaría expresar mis agradecimientos a Dr. Víctor Gallardo para la invitación aquí hoy y a Jesse Osabel y a la Fundación Sloan y todos los grupos que son participantes en el census de vida marino. Y actualmente voy a hablar en inglés. Es un poco más fácil, tengo mucha información que gustaría compartir con ustedes, entonces voy a cambiar. Y si tienen preguntas, estoy listo y disponible a responderlos y después de mi charla o esta noche. Gracias. Bueno, my name is George Shillinger. I'm with Standford University, the Center for Ocean Solutions, and I've been a participant in the tagging of Pacific pelagics program for many years, almost since its inception. I'm going to provide today, just present my outline here, a quick background on the TOP program and describe some of the work we've done in the North Pacific, our synthesis efforts, hotspots, and ocean observations, and then provide you with a case study on the species that I actually focused on, which is the leatherback turtle, and talk a bit about how our tracking efforts revealed observations about the influence of ocean currents on leatherback distribution and movements and implications for conservation, and then I'll give a quick wrap-up and describe next steps. And I just want to say up front that the TOP program is a huge collaboration involving many universities and researchers from all over the world. So the data I'm presenting today is data that's been created by a lot of different researchers from many different places. So first, just to sort of step back, when we look at the ocean from space, what we see when we look at planet Earth, we see a blue planet and a vast sort of featureless realm of blue that's not that much different from what we see from the deck of a boat or from a beach. A vast expanse of blue, and it's only with the advent of new technologies that we're really beginning to understand what's happening under this layer. And for many of us, when we think about for predators and prey, one of the images that comes to mind is the African savanna. And we all know from television and media and maybe our terrestrial heritage that where there's water, there's food, there are predators, and lions eat zebras. But when we think about the ocean, we have a more challenging perspective because it's really difficult to understand what's happening in the sea. So ocean predators present a lot of challenges. It's difficult to study ocean predators. You've got to catch them, you've got to handle them, you've got to find them. And so this is all relatively new field. And what we use to unravel these mysteries are electronic tagging technologies. And what you see here are several different types of tags. A pop-up tag, a spot satellite position tag. Here's a pop-up tag on a tuna, a satellite relay data logger, a GPS tag. And we use different animals as platforms for the research effort. And to do this, we had to really understand a bit about the behavior of these species and their ability to carry different types of tags and to know what kind of data we were actually seeking. So a lot of this required innovations and collaborations with tag manufacturers and engineers and oceanographers and numerical modelers. But and then along the way, we made some great innovations, retrofitting tags that were originally designed for elephant seals and sea lions for use on turtles and sharks. And this type of effort comes with a lot of challenges, attaching tags to fast-moving animals. And this is a shot by Bruce Mate, who did a lot of the citation work with the Census of Marine Life. And how to catch, as I mentioned before, and tag things that are really toothy, that are aggressive and strong and tough to work with, like these mako sharks. So we set out with a series of objectives focused on developing technologies for tagging, integrating different disciplines, marine ecology, physical oceanography, engineering, and many others, elucidating animal behavior at the scale of the environment, trying to use animals as ocean sensors to enhance our ocean observation efforts, and then ultimately to take this data and apply it to conservation and management. I'm not going to go into too much detail here except to say that the structure of TOP was relatively complex, involved a lot of coordination, a lot of different working groups. This is an interesting, should this program be expanded to other places of the world, it could take on different forms, but what we used in the North Pacific was organismal working groups, oceanographic integration groups, data management teams, and an education and outreach component. And we also worked with with NOAA and other institutions to figure out ways to serve up the tracking information live and integrate it with oceanographic data. And you can find this online at the Tagging of Pacific Pelagics website. And what you see here are a lot of tracks of different animals, and sea surface temperature, chlorophyll, and other oceanographic variables are served up alongside them. So this is a summary of all of the positions in the TOP data set. This data here is soon to be published, actually in Nature, and what you see are aggregations of different the tracks of three different species of tuna, elephant seals and sea lions, 3 different species of sharks, several different species of birds, loggerheads and leatherbacks, and whales. And color-coded there so you get a better sense of what's happening. And then just to quickly summarize those, the colors up here describe the different species. And what we saw as an overview is extensive ocean coverage, entire basin coverage, niche separation among species, 2 distinct populations of sea turtles, 3 different species of tuna with seasonal overlap and trans-Pacific migrations, including migrations like these bluefin here. And that seabirds covered vast amounts of ocean space in relatively short periods. So over the course of the last decade, we deployed tags on 23 species, over 4,300 tags, collected more than 1 million profiles, and are at 335,000 plus total tracking days. And the data we've derived from this work has been position data, species identification, temperature, light, pressure, salinity, fluorescence, and time series data, among others. And we've had great success using elephant seals as gliders. This is an image by Dan Costa, whom many of you may be familiar with, who's worked in the Southern Ocean for many years. And this shows temperature slices taken by elephant seals tagged in the California current. And we also are able to capture conductivity information from elephant seals as well with CTD tags, from which we can derive salinity and examine variability in the density structure of the upper ocean and at high spatial and temporal resolutions, and understand where masses of water are. And then what we came to basically elephant seals sort of emerged as the sort of imminent oceanographer in our animal tracking data set. You release what you see here is a track from a turtle tagged off, elephant seal tagged off Santa Cruz, and over 6 months it basically went deep into the North Pacific, collected over 10,000 profiles, and you can see here a 6-month window from an elephant seal's eye view of the ocean as the seal moved along this track. And what we learned with elephant seals is that they're prolific divers. They show great track fidelity. They go back and forth from foraging habitats and back into the California current system, and they segregate as well. So we have females going out heading out into the transition zone and up into the Bering Sea, and males heading north along the California current system. And seabirds were great monitors, or ocean sentinels for us as well. As I mentioned before, they covered a tremendous amount of terrain and took a lot of samples, as did tuna. And this is a tuna track that spanned over 600 days, tagged off of Baja. It crossed the Pacific and came back again. So what we've begun to discern then is how closely related species use the ocean differently in space. And what you see here are frequency of observations on the y-axis and sea surface temperature on the x-axis for laysan and black-footed albatross that are sympatric breeding birds tagged in the northwestern Hawaiian Islands. And they showed partitioning out in foraging during the non-breeding period in seeking ocean resources. So we'd see black-footed albatross is heading into the California current system, and Laysan albatross is heading into the transition zone. And similarly, we also saw some neat separation with bluefin and yellowfin tuna. And here you see bluefin tracks in blue, yellowfin in red, but there's also a lot of overlap in the southern part of the California current off Baja, where we were tagging both species and albacore at the same time. And with sharks, over here we have salmon sharks, which would make large migrations from where they're tagged in Alaska down the California current out towards Hawaii, moving from north to south and back again. And white sharks and these black tracks heading out towards the Hawaiian Islands and out into the Central Pacific into an area which we call the White Shark Cafe. They seem to spend a lot of time here. We're not exactly sure what they're doing. And that's a mystery that we're working on resolving now. And then mako sharks as well, who all three species overlapped, but they also showed very distinct temperature preferences. And we also were able to examine the correlations between various environmental variables and movements and see that during specific periods, you'd find all species together and then they would separate. And so from this data, we're starting to ascertain the presence of ocean hotspots and also examining regions of higher biodiversity. And we found that this Southern California bight and the California current region that sort of spans up towards into the Oregon coast, and with some key areas being the Gulf of Farallones here, the California bight, and also actually Baja has shown up. The Baja Peninsula as an important region as well, are important sites for both biodiversity and aggregation. So some preliminary synthesis conclusions. We've seen basin scale migrations of top predators. I've pointed out several of these to you previously. We've seen habitat overlap and niche partitioning among different species. This is the example of the albatross here and the sharks here. And we've identified key habitats as well, including these this transition zone region, these eddies that are propagated off the Gulf of Alaska, which are frequented by elephant seals, the California current system, and the White Shark Cafe. All of these areas have shown up as quite important. And what's also been particularly useful for us from a tracking point of view, but also interesting from a biological point of view, are these is the track fidelity we've seen both to migration pathways and to back and forth from breeding areas to foraging habitats. And here you see E-seals, and then up here, and these different colors represent some of the different species I've mentioned previously. So now to quickly move to leatherback turtles. There are two different stocks in the Pacific. There's a western Pacific stock and an eastern Pacific stock. Our tracking data showed that there's very little mixing. The status of leatherbacks in the Pacific is quite dire. As many of you probably know, they've experienced over 90% declines during the last 20 years. Our hope is that by understanding where they go and what drives their change and affects their behavior, we'll be able to figure out better strategies for protecting them. We used satellite relay data loggers produced by the Sea Mammal Research Unit. This is the nesting beach where we did the tracking work. We did three different seasons of tracking here. This is basically the last this is the last remaining high-density nesting colony for leatherbacks in the eastern Pacific, and this is off northwestern Costa Rica. It's called Playa Grande, and numbers there have declined. Nesting turtles have declined precipitously. This is our tracking data set over 3 different years. 2007 is in green, 2005 in purple, and 2004 in orange. And as you can see, there was extensive movement into the South Pacific Gyre with some movement into the EEZ of Chile. And there are, of course, reports of interactions with fisheries in Chilean waters as well. And quickly, just in addition, the great bonus from this work was that not only did we learn about the large-scale oceanic movements, but we had a great opportunity due to the turtles' and their nesting behavior to learn about what they're doing within the economic zone of Costa Rica and around the national park. And this is where they're protected, where they breed, but over the 3 months of internesting, turtles move extensively into unprotected waters where they're at risk from all sorts of pressure. So currently we're working with the government of Costa Rica now to figure out ways to apply this data to improved zoning. We also discovered elucidated the presence of a migration corridor for leatherbacks, which had been posited by Moriali et al. In 1996 in Nature, and he had attributed a number of different potential variables that might influence the creation of this corridor. We are in analyzing zonal currents, and there are several strong zonal currents that run through this equatorial region. We were able to determine that turtles actually increased their movements as they moved off the nesting beaches through the zonal current system. And I won't go through each of these phases, but basically we found a persistent migration corridor, which was published in PLS Biology and was subsequently used to create an IUCN resolution. Although it's just a resolution, it has some merits to work to protect turtles and mitigate fisheries interactions on the high seas. We stripped away the current effects and found that there was a directed southward migration, and we found that turtles really had two phases of migration, one in rapid movements through the equatorial zonal current system, and then slowing down into this oceanographic, what we call an oceanographic cold spot in the central South Pacific gyre, where you have an otherwise totally oligotrophic quiescent region, which makes us wonder what turtles are doing there. We know they're foraging, but it remains a mystery, specifically what food they're or what exactly they're doing there. You would expect that turtles would be associating with more productive regions along these shelf habitats, but instead they're going out into relatively unproductive open ocean areas. So that's a mystery we're working on as well. And finally, we were able to take this data and for all the work we're doing at sea and around the nesting beach, the leatherback situation is still dire, and turn it into a great communications exercise called the Great Turtle Race in 2007, which we found sponsors and drummed up a lot of interest in sea turtle conservation. And I won't, I think time is too tight to show you the race, but we're able to attract tremendous international attention to the cause of leatherbacks conservation. So finally, just in summation, I'm going to go quickly through this. I believe what we've created is a great opportunity to use animals as ocean sensors and sentinels, and that here at this meeting in particular, I just wanted to maybe put forth the idea that something like this could be carried on a grander scale in the South Pacific, and we've heard in many talks how important this region is, and how critical it is to have ongoing monitoring programs and integrate them. So as part of this effort, it's conceivable that this type of a project could be linked with the Dichado Ocean Observation System and tied to other marine life observing networks and ocean observing systems. And here's where we are today if you want to visit the GTOP website, and I want to thank all of the sponsors and supporters who've made this project possible. And that's it. Thank you.
Thank you very much, George. If there is any question, Jesse?
George, which are some of the first species that you would like to observe if you were to begin a program of tagging, an enhanced program of tagging here in Chile?
I think leatherbacks, swordfish. I would look at some of the seal populations as well, and probably, you know, what's really nice is that the California current work is a great corollary for the Humboldt current system. So you could use a lot of very similar species here. In fact, you know, the same species in many cases, but get a better sense for their distributions and movements in the South Pacific. But for sure, I think for me personally, the interaction between leatherbacks and swordfish is a fascinating one, both here and in the California current system, where we have lots of issues with drift gill netting efforts and possible closures and management of high seas habitats. And whales, of course, would be a great target as well. So seals, whales, and turtles, and swordfish.
George, I have a question. I think in Chile we would be happy to apply this technology to jack mackerel, to the Chilean jack mackerel. I see that most of the populations that you use are small populations. The jack mackerel is very abundant, at least in relatively compared to the species that you use. Could the technology be used to follow the migration patterns of jack mackerel?
Yes.
This is a big problem right now in Chile. It's a big social problem. The government approved large quotas for this year, I think 1,300,000 tons. The industrialists prepared for that kind of catch, but they were not able to catch more than 300,000. So there's a lot of people being fired, a lot of boats are standing. And so to predict this kind of thing in the future, I think would be very important. And everybody says that there's only one population, but perhaps these populations have different behaviors, like the ones of the tuna, for example, from the Gulf of Mexico and the Mediterranean. So these technologies would be very important to apply here very soon. It's urgent.
I agree, and I actually saw the article, a front page article in the paper this weekend on jack mackerel on this very issue or I guess it was Monday. And my thinking on that is that through a collaboration with Post and OTN, one idea, and this is something that obviously is years out, but that it could be developed, or the positioning of arrays along the coast of Chile, much as along the lines of what Ron presented earlier in some of these other talks, and so we could do a small acoustic tagging on jack mackerel, and eventually I think the tagging technologies are going to be sophisticated enough, and the miniaturization will progress enough that we could put pop-up tags as well on much smaller fish. So it would be really interesting to tag fish at different levels in the food web, and predators at different levels in the food web, and they would be a great target species. And actually, there's the Biologging 4 symposium that's coming up in March in Tasmania. We're involved in a workshop where we're going to be talking about tag miniaturization and how we might do these types of things. But certainly arrays and acoustic tagging would be a good way to do that.
Thank you, George. Is there any comment or question over there?
A question. This kind of program have a cost very high, I think. Do you have some experience with initiative like adopt a turtle and follow them through your webpage, something like that, to reach private donors?
Yes. Okay. So, may I show one more slide? I'm just going to. I might as well just show you this really quickly. This was what we did with one of the beauties of the tracking work is it really helps, I think, the general public get a window on what's happening in the ocean. And because of the charismatic nature of these species, they draw a lot of attention. And if this works, maybe it's not going to work. Shoot. Well, sorry. So these were the turtles in the Great Turtle Race, and we found different sponsors, corporations, foundations, nonprofits, and schools to underwrite the cost of a tag, and it also provide a premium for conservation on the nesting beach. So, and then when we ran the Great Turtle Race, and this is just one example, we also found an American comedian named Stephen Colbert, who we named the turtle after, and that attracted a lot of attention because he featured it on several of his television shows. But as a result, within 10 days, we had over 700,000 unique users visit the website we created, which had a lot of educational information on conservation and the status of leatherbacks and management of marine habitats and tracking data in oceanography. And we, over this period, we had 700,000 unique users, 3.5 million page views, and 70,000 people from all over the world signed on to learn more about sea turtles. So we provided content. When there were some lessons learned, the site was sticky. We set it up so that it would update every 10 minutes, and that wasn't actually initially the plan. It was just sort of something we happened along. And we found that people were texting in from all over the world and coming back to the website to see how the turtles were doing. And they've developed emotional attachments to different individuals, and we created cards with information, with baseball cards, we call them, for the different turtles.
Okay, thank you, George.
Okay, thank you.